All right. Sorry for the few minute delay. We're getting a few last things figured out. That's Okay. So what I'm going to do here is just sort of start by introducing myself. My name is Chris Malinowski, and I'm a second year PhD student at Florida State University, and I'm studying reef fish ecology with Doctors Felicia Coleman and Chris Koenig, with Dr. Felicia Coleman as my major professor. And so, before I get going into anything, I really wanted to thank Teens for Oceans and Explore.org for hosting this chat and for allowing me to talk about my research and overall interest in reef fish ecology. And so there are a lot of fantastic people actually working behind the scenes in Teens for Ocean and Explore.org, a lot of whom I've met with and spoken with over the last few weeks. So I wanted to really thank them for all the effort they put into this. And so I want to get into my background a little bit and just give you sort of a little bit of information on how I got my start, sort of what my interests are and how they developed. And so I'm originally from the cheese state of Wisconsin, and I moved to South Florida to complete my master's degree at Florida Atlantic University, studying foraging ecology and in spotted dolphins and bottlenose dolphins in the Bahamas. And then I moved to North Florida in pursuit of my PhD at Florida State University, and am now currently in my second year. And so, yeah, and just a little bit about my interests in aquatic and fish ecology. And again, as I give this sort of intro into sort of my background and research, you guys are free to email questions, send questions, and I'll try to answer them as I go along too. It's sort of an informal thing that we're doing here. All right, so I've always been interested in water and fish pretty much since as long as I can remember, but there were some definite experiences along the way that really cemented this for me as a career path that I wanted to pursue. And so I spent an undergraduate semester in Australia, which I spent a lot of my time diving on the Great Barrier Reef, which were some of the most beautiful reefs I've ever seen. Really sort of struck me as something that I really wanted to work with in that system for my career. But even probably a more pivotal point for me was my research experience for undergraduate, or my REU internship that I had at the Great Lakes Water Institute in Milwaukee, Wisconsin, where I lived for quite a few years. And so during that time, I worked with Dr. John Jansen on a variety of projects related to fish ecology and also anthropogenic impacts, which have a lot of I think they have a lot of impacts on a variety of species, especially native species in the Great Lakes, Lake Michigan in particular, where I spent my time. And also invasive species, and I see that I'm getting a couple questions on lionfish already, which I'll get to because I definitely have some experiences with lionfish in my research that I can discuss in more detail. And so beyond the Great Lakes, continuing with my background, I've also spanned a lot of moving beyond the Great Lakes into a lot of other wetland and marine ecosystems, which have included the Bahamas, like for my master's work, the Everglades, and of course the Gulf of Mexico and South Atlantic, where I'm currently pursuing my PhD dissertation research. And so if there's one thing that I've noticed most in my relatively short time in this field, it's that we are in dire need of more conservation-minded practices on all facets of life, but of course I'll focus on fish for this talk, since issues facing our fisheries are certainly some of the most dire that we're up against in upcoming generations. And so before getting into this main topic of the live chat, which would be my research and things about reef fish ecology, I wanted to briefly mention the importance of efforts like those of Teens for Oceans and Explore. And so these two organizations do incredible work sparking interest and gaining awareness through exploration. I mean, if nobody cares, then who will fight to protect our oceans and the organisms that depend on it, including us? And so for me, curiosity and concern for the oceans are probably my biggest drivers. I have a feeling that many of you who are actually tuning into this live chat today have very similar interests, otherwise you wouldn't be here. And so the importance of such awareness and curiosity is not only for developing more scientists and for the purpose of scientific advancement, but also for public awareness of issues facing our oceans. And so bringing these live underwater webcams is not only cool, but it also allows people, especially those who are landlocked, like in Colorado, where Teens for Ocean is, to view and for students even participate in research projects, which is really cool. And so part of why I appreciate what Teens for Oceans and Explore are doing is that at FSU CML, or the Florida State University Coastal and Marine Laboratory, where my lab is located, we also strive to create many excellent programs and opportunities for younger students to get hands-on experience, which many of us graduate students that work there actually participate in, including me. And so even for my research, which I'm going to get to here very shortly on Goliath Grouper, I'm hoping to involve more students in what I'm doing and through my research. And I've recently even discussed the possibility of working with a student with Teens for Ocean to potentially sponsor student involvement in some aspect of my research, which would be really cool. And so for any students, high school, undergraduate, and even middle school, who are listening, if I could offer one piece of advice to you in doing something for yourself, it would be to get as much experience and exposure to aid towards your interest in science or any field as possible. Get as many hands-on experiences or exposure to things as much as possible. I mean, nothing truly beats hands-on experience for discovering your true passion about something and figuring which path you should take. And so those of you who tune in often to the the webcam locations like the Cape Fear one that should be associated with this broadcast, that counts as really good exposure. And I encourage you to watch, think, identify, even write about what you see. I mean, I'm often, every time that I go diving or look at reefs, I've got a reef identification book with me. Some of them have nice pictures. Other ones that I use for species that I'm not as familiar with, I'll use dichotomous keys and really try to figure out what it is that I'm looking at. And so, just as I'm talking here, I'm going to keep looking over to look at some of these questions. All right. So I'm getting some questions about the camera itself. One thing that I wanted to mention is that, as I'm looking at these videos, I've spent a little bit of time this morning, and one thing that certainly jumped out at me while I was watching some of these live cam feeds were the differences in species assemblages and diversities between all of them, but also, knowing that different habitats and regions differ but have many similarities, so which is true of reef fish in general. So they, from the Great Barrier Reef to the Red Sea to the Caribbean and Gulf of Mexico, there's reef fish species that are associated both with coral reefs and, as I mentioned, even that aren't necessarily associated with coral reefs but have many reef-like features to them. So there's a lot of similarities, especially in family diversity, maybe not as much with species diversity when you move away from the Great Barrier Reef. So reef fishes have actually been around for a very long time. Many different definitions have been proposed for reef fish. And for the purpose of this talk, I would define reef fish as those taxa that are found on and are characteristic of coral reefs but are not restricted to coral reefs. And so what I mean by that, and what I just mentioned before, is that most characteristic reef fishes or fish families do not disappear when coral reefs stop, although species diversity may lessen. All right, and so we also believe that the history of modern reef fishes began sometime around the early Tertiary, so about 50 million years ago, with origins spreading back to at least late Cretaceous, 70 million years ago, and possibly even the early Cretaceous, like 100 to 130 million years ago. And so they've been around for a really, really long time. And of course, a lot has happened since then to get to where we are today. But what is it about recent human civilization, and specifically the last few decades, which has gotten our fish and fisheries into such a perilous state? Many are overfished, even leading to local extirpation or extinctions for some species. And so some of the major culprits are development and other means of habitat destruction, pollution and eutrophication, so too many nutrients from land-fed sources going into the ocean and streams and rivers and pretty much every aquatic environment. New technologies that have led to overfishing like GPS and scuba diving, and overall poor resource management. And so all of these things together have really sort of gotten the species that I'm going to talk about in my research with Goliath Grouper into the state of peril that they're sort of in right now, even though they're showing signs of recovery. And so before I get into that, I think the thing to do is to maybe answer one of these questions. Let me see here. So let me just read this one. So about lionfish, are they prevalent in the Cayman like they are in Florida? And what can we do to stop them from growing in population to protect the reefs? And so lionfish have pretty much spread throughout the Caribbean. We're seeing them just as of recent in some of the very deep waters that we're exploring in the Gulf of Mexico, the Atlantic. Yes, they're extremely prevalent there, and they're certainly spreading out. There have been, a lot of management ideas, and in particular, the biggest thing that people are trying to do is have these lionfish derbies and pretty much spend time diving and spearing lionfish on reefs. Maybe it makes a dent and has some current impact on it. But there's really, unfortunately, at this time, no real way to stop the spread of these fish. They're extremely good generalists, and they feed on a lot of things. They reproduce very fast, and they seemingly have no real natural predators in this invasive environment, coming a long way from the South Pacific. So, yeah, short answer to that is unfortunately no. There's no real thing to do to stop the spread, as far as we know right now. So hopefully further research will give us some more ideas of what we can do to do that. But any impact of divers even going down and spearing lionfish and trying to keep them at a minimum on reefs is certainly beneficial to the ecology of these systems. What changes in the population and the viewing experiences can we expect as seasons change, or won't spring, summer, fall, winter matter here? So yeah, that's a good question. So that was from Valley Flowers. And seasonal changes, yeah, they vary quite a bit for some species. A lot of reef fish species have spawning aggregation movements where they move around depending on the season. So, when I'll get to Goliath Grouper, for instance, they have a very narrow window of aggregation time from about the middle of July leading to about early October, where they're prevalent in mass numbers on a small number of sites, and then dissipate back to their native, or not native, their resident areas for the rest of the year. And so season matters big time for a lot of reef fish. Some are more active during spring, summer months, and less active during the fall when it's cold out. So there's a lot of different reasons why season would have an impact on what you would see when. All right. So before I get to any more of those questions, maybe I'll give now a brief introduction about my research project on Goliath Grouper, and then sort of talk about some of the objectives of what I'm trying to study. So the main thing that I'm trying to do is evaluate mercury levels in Goliath Grouper, and to understand when, where, and how it is impacting their health and potentially their reproductive success. And so what do I mean by that? So where is it coming from? So mercury, in particular methyl mercury, is most commonly accumulated into tissue in these fish, or anything for that matter, by ingestion, so consumption. And so in doing diet studies and analysis of Goliath Grouper, which we've been doing for some time, we're able to understand better what the dominant players in their diet are. And then looking at that and taking those, and we have actually preserved a lot of those samples from their stomach contents. I can look at mercury concentration in whole prey of some of their dominant prey, and actually start to understand what's impacting these fish and where they're getting most of their mercury from. And so that's a big question we want to know. And also, I'm looking at, is it regionally different? So typically all fish are not the same, even though they're presented that way when you're giving like hazards of consumption limits of fish to eat. But there's a lot of differences in the environmental conditions of where fish exist during different parts of the year or inhabit. And so I'm really interested in looking at, different areas these fish are coming from that I'm collecting during the spawning aggregations, and what regional differences there may be that are dependent on environmental conditions that are making mercury and other toxicants more bioavailable to these species, and then consequently more uptake and assimilation into their tissues. And so when, the question that we just talked about of whether or not things change seasonally, well, so in these species, there's a lot of changes that occur, especially during these large movement patterns, which we know from our acoustic moving data or monitoring data that they can move up to 300 miles, or at least that's the maximum that we've seen in the species of 50 that we've tagged with acoustic tags. So moving basically from South Georgia down to these spawning aggregation sites off of Martin and Palm Beach counties in South Florida. And so they have these large migration patterns, as a lot of large predators do. And so what impact does that have? Are they getting more of their mercury or other toxicants during times when they're away from their residential areas and they're in these large spawning aggregations? Are they feeding more then versus non-spawning times? And so are they getting more mercury during these times, et cetera? So one might have a big player, might be a big player in this. And also, how is it impacting their health and reproductive success? So their health, I'll be measuring and have started to measure various health parameters and trying to understand how this might be impacting their immune system functioning, oxidative stress, and overall health status, and also reproductive success. Are females offloading mercury into their eggs? Is that having an impact on overall reproductive success and egg viability, so that they go off to hatch and live and be healthy and add back to the population? These are all really important questions. And so they're especially important for this species because Goliath grouper, as many of you may know, are considered globally as critically endangered by the International Union for the Conservation of Nature, or IUCN. And they've been legally protected from commercial and recreational fishing in the U.S. since 1990, when overfishing caused a sharp decline in population numbers to the point where fishermen even went to councils and asked for these fish to be listed because they saw the numbers drop so quickly in a very, very short amount of time, like on the matter of a decade. And so there's also some behavioral and life history characteristics that have sort of worked against Goliath grouper. One being the obvious, which is that they are large and long-lived, which means that they have a long time to mature and to reproduce and add back to the population. And also, because they're large and long-lived, they have a lot of time and accumulate a lot of mercury and other toxic substances into their system. And they also have high site fidelity. So I mentioned these large migrations that happen where they come to these spawning sites during part of the year. But the rest of the year, as most grouper species, they have very strong site fidelity, meaning they stay in one place and don't move much very far from that during most of the rest of the year. And so that makes them very easy targets to fishers to go and, especially with the advent of scuba diving, to go and basically hit these things, which are like sitting ducks basically, and it makes it very easy to overfish them. So one good thing is that the Goliath grouper, since their harvest closure in 1990, they've responded very encouragingly to protective measures, but their actual recovery status is very uncertain at this point. And given the slew of issues this species facing the species in their recovery, I would suggest that we need to exercise extreme caution in any discussion of reopening this fishery on any level, which is, by the way, currently being considered. Maybe look back to see if there are any questions as we're going. Do you have any more questions? Okay. Doesn't look like there are many more coming in, so let's see here. All right, so I've got one from Southern California saying, Chris, I would like recommendations on IDing the fish we see here, especially any online links. Thank you. So any information on that, I would say FishBase is a really well-known taxonomic identification, which has all the current listings of species. Maybe not so good for actually comparing pictures and identifying things. The best way to do that, I would say, is probably getting fish identification books. There's a lot of them out there, some better than others, depending on what level you're at. So I mentioned dichotomous keys before, which would probably be a more upper level way to identify fish, where you actually count things like fin rays and other things when you've got fish up close and personal. But the best way to do it is probably just from image comparison. I've got this book right here that's a pretty good one, Reef Fish by Paul Heumann and Ned Deloach. That one's pretty good. It's got really easy to see pictures in it that are fairly representative and short descriptions. There's a lot of other ones. I probably have somewhere behind me, but yeah, I would my advice would be to get some go and buy a few books, probably, because nothing beats having things in front of you when you need them. If you're a diver or you're looking at these on the camera, having something in front of you would be good. As far as websites go that have good identification, I don't know of any good ones, other than just searching species, because the best thing you can do is page through pictures if you don't know what something is. Yeah, when you're out in the field too, there are also some good waterproof books. I'm not sure what the names are. You might have to look them up, and cards that you can have just for easy identification. Yeah, so that's my advice with that. I was wondering if somebody's going to notice that. From Myrtle LTJE, she says, being a fossil collector or a fossil gatherer myself, must say I'm very jealous of your beautiful ichthyosaurus in the back, but my question is how many times and in which periods of the year do fish reproduce? Yeah, so I've got this, if you can't see that behind me, the ichthyosaur, it's just a mock ichthyosaur. They're actually much bigger than that, which for those of you that don't know, they're around hundreds and hundreds of millions of years ago, like 600 millions of years ago, and they're related to reptiles, even though they basically evolved convergently with dolphins or look to be a lot like dolphins and other marine mammals. So, yeah, good identification. And so, in which periods of the year do fish reproduce? So that varies very much on location. So latitudinal gradient, even within species, that varies between species. Yeah, it's very variable, extremely variable. For most reef fish species, spring, early fall through early fall are the main times, at least in the northern hemisphere, where a lot of fish spawn. But again, that varies depending on species. So I'll keep going back and forth with the questions. Let me keep moving along with talking about my project as we do that as well. So one of the big issues with understanding the biology of Goliath grouper is there still remains a lack of basic and baseline biological data. And by baseline, I mean before these things were basically overfished to near population collapse, or so we may assume. There was not much known about them. There's no real fishery dependent data that was collected, and so now after the fact, we're trying to collect as much fishery independent data, meaning through research, that we can possibly get in. And so there's a lot of people that have been doing that, particularly our lab, and Chris Koenig and Felicia Coleman have put at least over a decade of work into trying to understand the biology of these fish, from life history, demographics, movement patterns, and a slew of other things. But there still remains a lot not known about them, and so that basically encourages this project and in trying to get as much information on their age and growth patterns and movement patterns, a lot of things that we need to know about these species in order to protect them. And so this lack of basic and baseline biological data, which includes a lack of reproductive and age structure data, which we've got a grant to currently be working on that. But it makes it difficult to assess the level of recovery of these species without having a complete set of data on that. We also know that juvenile habitat is in decline, which really works against them. So through development and destruction of things like mangroves, which we know to be the primary juvenile habitat for this species and a lot of reef species, with that being in decline, even if we protect the adults, protecting, not having habitat for the juveniles to grow and develop and contribute back to the population is really sort of works against their population recovery as well. And so, in juveniles, they spend about the first 6 years of their life in these mangroves before they are even able to mature and move offshore and start to join the adult population and become part of the breeding population. So, I'm talking again about their long-lived nature and that and their strategy really can work against them if they're being overfished. All right. So let me look back to see if there are any questions. how do you catch the groupers you sample? So I guess I can talk about that now before I get into a little bit more detail about some of the things that I'm seeing in my preliminary analyses. So what do we do? So basically, these fish are sampled in a number of ways. Some of them, part of our work is actually through diving, where we actually look at their community assemblages and abundances and habitat association using scuba. And so it's completely hands off. We basically do a measure, an estimate of their population size, and we measure the fish. So that's a big part of it. So, get to the other side of that, which we catch them with hook and line and bring them onto the boat and take samples. But in the water, one of the important things is that we understand whether or not we have a representative sample set of the population that we are actually surveying. And so counting the individuals and especially getting size estimates gives us an idea of whether or not the fish that we're catching on the boat and taking samples from are representative of the population. And so technology has really sort of taken off and benefited underwater survey in a lot of ways, one being compactability. So cameras, I think one of my profile pictures for this was me holding a very large Sony camera, underwater camera, which was about probably, at least 10 times the size of this little GoPro camera now that I can take down and do the same thing with. And so what we would do then to get a size estimate underwater of these fish is actually position two lasers on either side, and which is at a standard rate. Usually we have it at 10 centimeters separated, and if we get a perfect shot perpendicular onto the fish as it passes, we can take it back and basically estimate, knowing that size of those 10 centimeters, we can estimate the size of the fish back in the lab. And so that gives us a really good size estimate to compare. That was a really good question. And so on the boat then we use hook and line, right? So we basically bait these very large hooks to give you an idea. I actually have one here that was broken off. So we've got this very thick monofilament, much different than something you'd catch a smaller fish with. It's probably about a 2,000 pound test, which is huge. And then this hook, which I had a feeling somebody was going to ask this question, so I thought I'd bring something for at least a size reference. So you can see how big this hook is. I don't know, this was at about four inches or something like that across. So it's a fairly big hook. It's the biggest hook size you can really get. It's called a 20-aught hook. And so this is what we use to catch them, basically. Put a big hunk of meat on here, and while they're spawning, they basically have these large aggregations around artificial and natural sites, and they'll catch onto this hook, and we'll pull them onto the boat, and basically put a towel over their head and or over their eyes, which basically protects them from any damaging effects of sun rays. We put a hose in their mouth, pumped from the sea, so that water and oxygen run over their gills, and then basically we take our samples for a number of different research objectives. And just to also give you an idea, it's really kind of cool to see the power of these animals, which is enormous. For when it goes wrong and they typically dive back into the wreck. You can see this thing. This hook has been completely bent, which is a really strong hook, by the way, by these fish. They're enormously powerful. So that was in comparison to this one. So anyway, so hopefully that answers your question on that. Let me see here. So this question from IC, what is the long-term possibility of applying the results of mercury and toxins, toxicants? Toxins are produced in the environment typically, not human-made. So mercury and you mean toxicants in the Goliath grouper or in the Goliath to effect on mankind over time. So I'm not sure exactly what that question's getting at, but if I'm interpreting that correctly, you're talking about some sort of modeling scheme where we'd put mercury into maybe a system and look at how that affects people over time. So that's sort of a difficult question, and so that's sort of a mixture of looking at human health and fish health, which really you need to understand fish health, or the things that we're consuming in order to understand its impact on human health. And that's not so easily answered of a question. A lot of what we know about the effects of mercury on human health are probably more on a short-term scale of, generational. So if a mother consumes something that's very high in mercury, she might suffer the effects, be it neurological or other effects, and then potentially pass it on to a developing child, and have a range of effects from developmental issues to other neurological, you name it. There's a ton of different effects that mercury can have. So there's no easy answer to that. But there's definitely a lot of health research out there on humans that I'm not as aware of how this is impacting the human population. But we do know that it's not good, and that there's a lot of issues that are associated with mentally, neurological, with mercury. And while we're on that topic, let me just add something about my preliminary results from the study, because I think they're really important, and we're onto something big here. Because the values that I've measured, on just a small sample set of what I've collected so far, still have a lot to be processed yet, but the mercury levels are extremely high. I mean, they're likely the highest reported for any grouper or saranid, which is the family that groupers are in the Gulf of Mexico or Atlantic Ocean. So even of all the other studies on groupers that have been done, of the 10 white mussel or fillet samples that I've looked at, in the 10 liver samples, they're higher, if not just as high as the other samples that have been, and mercury levels that have been reported in the Atlantic and Gulf of Mexico. So that's already pretty big, and so we need to really understand and figure out why that's happening and what that means to these fish. And so just for reference, so the levels that I've measured, on average so far for average liver mercury concentration has been 7.6 micrograms per gram. For fillets has been 1.9 micrograms per gram. So just try to keep these numbers in mind. And for maximum liver, it's been 31.6, and for mussel has been 3.6. And so for reference, the typical human consumption limit warnings for mercury contamination in fish is 0.5 micrograms per gram. And so just to put that on scale, so 0.5 is the maximum by most standards, and I'm seeing it as high as 31.6 in the liver. Of course, we don't eat liver, so we're more concerned with muscle tissue, but a maximum of 3.6 is about 7 times higher in Goliath grouper than the safe human consumption limit that is set by EPA and other standards. So that's already a huge thing that could be impacting these fish. And certainly, if we're considering opening this fishery back up on any level, that's a huge concern. I mean, I certainly, for one, would never eat this fish. And so definitely something to keep in mind as we're having these discussions. Yeah, and so, in talking about Goliath grouper recovery and the recovery potential, this is certainly having a big impact on that. To what extent and what we can measure is going to depend on what we're able to show in some of this research, but certainly it's having effects. But yeah, it's a matter of what we're able to measure and then correlate with this mercury exposure. Okay, let me go back and see what kind of questions I've got. Let me scroll down. How much does a full-grown Goliath grouper weigh? Well, that's a good question. I also assumed somebody would ask that at some point. So the Goliath grouper actually they're the biggest teleos, meaning bony fish, in the Western Hemisphere. And so they get up estimates of the upper ends of 800 to 1,000 pounds, which would be very big. I think, typically the ones that we catch are probably closer to 400 pounds, maybe up to 500 in some of the bigger ones we've seen. The oldest one that there's actually validated evidence for is 37 years old. Of course, we're not seeing those in the population that we're studying at this point, because they've only been protected since 1990, and we also know from some work done on their aging data by Deb Meary and others at University of Florida, that a majority of them have come back into the population since about 1999 or 2003, since 9+ years after the actual closure and harvest closure was put into place. And so they average from for females in our population so far, for the ones that we've looked at, range from the ages of about 6 to 19, and males 5 to 16. Of course, those are the mins and maxes. The majority of females were actually between 9 and 12 years old, and males 9 to 10. So we're still looking at a fairly young population. And the scary thing, I guess, is that mercury levels we're measuring are very high already. And the longer a fish lives and the bigger it is, the more likely it is to bioaccumulate these things. So it could be a complete hindrance to their population recovery. But I guess that's a lot of the question that I'll be answering with my research. And so I hope that answered your question. So they get really big. There's not many other species that really pair with this one in terms of size. There's actually a subspecies which used to be, which was, I guess, before considered to be the same species as Goliath, is now considered a separate species in the Pacific, and they're probably on average about the same size. And then there's another species which is the biggest reported grouper in, I guess, the world, which would be the giant or the Queensland grouper in Australia, which there's estimates all over the place, but up to 12 feet in length, about the same weight. But in general, these fish they're very long, but they vary a lot in girth, and so without having an accurate measurement of their weight, length by itself doesn't completely do it. Let's see here. Let's look at some more questions. Could the findings of this research be used as a deterrent for fishers in the long run? My answer to that is definitely. So right now, with their protected status, they cannot be harvested, which means they cannot be brought onto the boat and or consumed. But they can still be caught. All right, so a lot of people fish on these reefs where they aggregate or where they exist, and either incidentally or purposely catch Goliath grouper, which is not illegal. So you can pull them up to your boat. You just cannot land them. Without getting on a tangent, which I was just about to go on, the answer to that is yes. So definitely I would not consider consuming this, and I would think that fishermen or fishers would strongly consider not consuming this fish based on the mercury levels that I'm seeing. There's also a report by Adams and Son in 2013 that showed very high levels as well. And so, and a lot of those were collected from the Gulf of Mexico. And so, we know the levels are very high, and I really think this would be a deterrent for most people. And if it's not, it should be. Let me see here. So there's another good question from Patty Moo. Policing the South Florida Yahoos, ESP, especially Miami Broward, lived here many years and have seen abuse of fishing and divers. Do you think this needs to be addressed? Yeah, I mean, it needs to be addressed. So there's a lot of abuse from people in general on resources. People are very interested in Goliath grouper. They're a huge fish. They're an attractant for a lot of people for both fishing, diving. And so one of the benefits of this, at least from an economical standpoint or ecotourism standpoint, is that one of the big reasons why Goliath grouper are being protected is now because of this huge dive industry that's been created around these large fish, which are really exciting to see. I mean, people are coming from around the world to see these fish now, and it's a huge attractant. We've talked to a lot of dive shops about the huge increase in diving that's occurred in the last couple of years since these fish have started coming back. And so it's basically started a huge economy boost for a lot of these divers and dive shops. So some of this is weighing the benefits with the costs of it. There needs to definitely be more awareness, and there's a lot of people pushing for that on the actual interactions that people are having with these fish that are on these reefs. Respect for animals is a huge thing, and in general, and for these fish that show some signs of curiosity when divers are around, definitely it needs to be addressed that people should keep their distance. It's a large predator, just like anything else that needs its space, and not harassing animals is one of the responsibilities we have, with being able to having the wherewithal to get in the water with them. And so that definitely needs to be addressed, and probably needs to be addressed more. As far as fishermen go, people want to catch these fish as well, and you can't stop them from doing that. You can keep them from harvesting and from controlling it. People are going to abuse that anyway. And certainly, poaching can be occurring. There's, with, even when enforcement's put into place. So, you can't stop anything. No system is perfect. But certainly awareness needs to be pushed. And so there's a lot of fishermen for these things that are actually very beneficial to the system as well. A lot of who we work with. So they're not all yahoos. Of course, there are a lot of them out there, but a lot of fishermen have actually helped us out tremendously with collections of data. So because of this, they basically are allowed to work within some confines of our permit, and they can collect samples for us and send them to us. And we've gotten tons of extra samples from people that have benefited from basically using Goliath Grouper to go on charters as a way to help with research and to also boost their business, which I think is fine. And so it as long as it's done correctly, and then the people on our permits are doing it correctly. So there's a lot of fishermen, some who are extreme proponents for conservation of the species. So we're definitely headed in the right direction with awareness of this right now, and certainly more of it needs to be addressed. Yeah, and so I've talked a lot about the Atlantic coast and the spawning populations there. Our research also expands into the Gulf of Mexico, and the Gulf is, since we can't be in all places at once during the year, we have targeted the spawning aggregations in the Atlantic, but similar things are happening in the Gulf of Mexico. Fishermen are helping us there. Angela Collins at FWRI did her dissertation work on looking at movement patterns, she's found similar north-south movement patterns, just like we've seen in the Atlantic side. So there's a lot of similarities and comparisons to be made there. So there's a lot of issues to address, certainly, and not just with Goliath Grouper, but with all organisms in the ocean, really, and certainly reef fish and grouper, since most sort of grouper are one of our biggest resources for recreational and commercial fishing. So I think that in particular, they need to really be focused on for a lot of protection. Most of them are overfished, and so we really need to start considering better management, and this is being done all the time. People are researching these things, so that's okay. So I sort of laughed at this question that I read at first, but it's actually a really good one. So somebody is asking, when you tell someone that you study Goliath groupers, what is their usual reaction? Do they express opinions about fishing them? Yeah, that's a really good question. So just like a lot of other, of course, charismatic megafauna, people have extreme opinions on what's going on with these species. They've heard a lot about it. They've got opinions going one way or the other. Unfortunately, a lot of times they're uneducated opinions, and which is why we're making this huge push to educate people on them. So it depends on who you talk to. Some people that have a strong interest, and there definitely is, in opening up this fishery, are very strongly opinionated and outspoken about how they feel these fish are taking over the reefs, eating everything on them. And it can turn, in those types of conversations, can make this a very controversial topic, which it's not. So Goliath grouper are native to the area. They were almost completely overfished to the point of a collapse. And now that they're showing some signs of a recovery, meaning that they're coming back on these reefs and in large numbers, which is how they used to, people aren't used to seeing that. So in science, we call this thing shifting of the baselines. And so what's basically happened since 1990, before that, when and compared to now, people that know the Florida system now that didn't know it, before these things were overfished, we're not used to seeing these large fish here, and now that they're seeing them, they're forming all kinds of opinions on what they're doing to the reefs. When in reality, they're having a very positive impact, and research has shown on the actual ecosystem. So, for instance, their ecosystem or ecological modifiers that, as far as we know, much similar to red grouper, so they have a very positive impact that way. So on a lot of natural and artificial systems, they actually they'll benefit the reef from clearing out areas, making more area or relief available to more species and higher numbers of species. So, for instance, on a site where there's a lot of sediment that would build up from wind or wave action, or from just sedimentation, their actual movement in and out, because they're so large, in and out of these systems actually clears out areas and makes it more available to many other species. And we've seen and measured actual increase in abundance and diversity of some species just based on their overall presence in sites. And so they have a huge benefit to the system that way, and they're native, and so yeah, they've always been here, and so they are part of the ecosystem, and we're just trying to figure out what exactly that was. And now comparing this to something like lionfish, which there were questions on before, which is invasive and is coming up in these huge numbers, that's something to be concerned with and to have an issue with, right? Because we don't know what they're going to end up doing to the overall ecosystem health. And so far, evidence and research has shown that it's very negative. They're eating a lot of, pretty much everything on the reefs, and diversity is going down, abundance is going down, but these species are very prevalent and increasing rapidly. And so, if we're going to be concerned with any species that's occurring in large numbers where they didn't used to, it would certainly be something that's invasive like the lionfish. But yeah, people are very opinionated about it, as I would expect them to be, and I think I would encourage opinion, but I would encourage it to be educated over just outspoken. Let me see here. So there's a question about what are the main sources of mercury contamination? Is there something we can do to lessen bioaccumulation of mercury in all fish species? So again, these are really good questions. Mercury in itself has a very complex dynamic and cycle in the environment. So a majority of mercury that's gotten into the environment has been industrial, and actually since the Industrial Revolution, there's been multiple estimates, but between 3 and 4 times the amount of mercury that existed before has been introduced into the environment since the Industrial Revolution, which is a huge amount on scale. And so what happens, and it's introduced in a variety of ways. So combustion of coal, mercury is typically found in a lot of rocks and core elements, and once things are broken or burnt, it's released into the environment where it was before stored. And so, and we have products that we've used mercury in before. We've gotten a lot wiser with that. Thermometers, even light up shoes. 10, 15 years ago, those LA gears and those shoes had mercury that was allowing that to happen. So they're matting hats. Mercury's been around for a long time, and we didn't always understand the consequences of that. But getting back into how dynamic and complex mercury can be in the system, we still don't know a lot about it. But basically, once it gets burnt and airborne, there's a lot of different forms of mercury. So elemental mercury can go up into the atmosphere. It can last there for up to a year, meaning that it can shift with winds with the atmosphere, and it can land pretty much anywhere. There's probably higher concentration of mercury that falls out near where it gets exposed back into the environment. But it's basically omnipotent at this point. It can be found anywhere. And then, of course, it settles out, and depending on environmental conditions, physical conditions, it has any number of interactions. It can settle out in the bottom, and depending on the microbial community that exists in areas, will determine basically if that elemental mercury or other forms get methylated. We talked about methyl mercury as being the most toxic form that we ingest, and that has all kinds of neurological issues. Well, that actually happens at a microbial level where it becomes methylated, it gets uptaken by, very small organisms, plankton, and it gets ingested in the next, if you'll call it a trophic level, which there's always discussion on what trophic levels mean. But as things get bigger and eat more and more fish, so by the time you get to top predators like sharks or, some grouper species, Goliath grouper are actually lower trophic level species, but they're long-lived, so they accumulated a lot. But the more something eats, things that have eaten mercury, accumulate a lot of it. And even mercury that's been in existence and buried for, decades could be reintroduced from dredging, or one of the big theories is the oil spill that happened. What happened? How did that impact the environment? Well, one of the big theories is that it basically created an anoxic environment, enabling this microbial community to remethylate mercury and potentially create a big boom in methyl mercury back into the environment. So, yeah, mercury can cycle and recycle, and then depending on its half-life, maybe break down eventually. But yeah, I mean, through less use and less combustion, those are the best ways to decrease our amounts of mercury. Let's go back to look at some more questions. So, do I have a favorite moment that I've had diving with Goliath grouper, or any diving moment? So many. Obviously, it's one of my favorite things to do. I love to dive. I think every time I've gotten in the water, I can still remember the first time just a few years ago when I witnessed one of these huge spawning aggregations on these wrecks and reef sites. It was one of the most miraculous things I've ever seen. Yeah, so I can still remember that one in particular. I've seen up to 100 plus of these large fish on sites before, and it's like nothing I've seen. And I can definitely understand the attraction for a lot of people to come from around the world to see these fish, and I don't blame them, because it's an incredible sight. And I've also seen lionfish in the Great Barrier Reef, where in their natural habitat, and I thought it was one of the most marvelous sights I've ever seen. And I still think they're beautiful fish, but it's obviously tainted by the adverse impacts they're having on the communities and fish communities and environment right now. So yeah, for those of you who aren't divers, I would encourage you to to get out and do that. But of course, these live video feeds are incredible too, and I've sat and watched them now for a while, and I'll continue to do so, because you're seeing things that technology has allowed you to now see, which was not available even a few years ago. So a really cool thing that's going on right now with Teams for Ocean and Explore, and I'm excited about it. Let's see here. Yeah, keep your questions coming. These have been extremely good questions. Is there any that I'm missing? So again, let me sort of one of the fun topics that we had talked about earlier about catching these fish, and we emphasize too that since they're protected, we've spent a lot of time developing techniques to sample from these fish non-lethally. And so, for instance, for aging, which has historically been done and still being done on a lot of fish, is the use of otoliths. So these little ear stones that are in the fish heads, which basically you can count sort of like rings on a tree to get an estimate of the age and growth patterns in these fish. And unfortunately, that technique requires the fish to be consumed or put down in order to do so. But with Goliath grouper, we're able to utilize technology to do the same sort of measurements using their fin rays. And so we can basically excise or take out a couple of their fin rays, in which they grow back, we know, within 6 months to a year, or at least their tissue mends itself and back over, and it's of really not much harm to the fish. And we're able to get the same estimate out of those rays. And so that's been really cool. And a lot of the other ways that we've been able to measure things, taking gonad biopsies with just a vacuum pressure tube instead of having to cut the fish open, is very minimally invasive to the fish. and usually within a few minutes we've sampled these fish and gotten them back into the water, and we know that there's a 98% survival rate of fish that we've sampled. The couple that we know that have not made it have likely not been because of anything we've done. There's one picture that I've got of a fish that had mounds and mounds and mounds of monofilament wrapped with hooks in its stomach and its mouth, and we spent some time trying to cut it out and getting hooks out, and we know that one probably didn't make it. One of the really cool things that that I wish I could show you a picture of, I don't have in front of me, but is taking stomach contents and actually getting those from the fish. And there's two main ways that we're able to do that. On some of the smaller fish, we'll use a lavage technique, actually pumping water into their stomachs, and they'll regurgitate their food. But in general, the main thing that we do is we put a PVC tube into their mouth to keep it open and to keep your arm from being basically clamped down on, and you stick your arm through the mouth into the stomach of the fish, feel around for food. We'll often find, or their prey, we'll often find hooks in there that we'll get out to help the fish that may be causing them some ailment, and fishing lead and fishing line. Yeah, there's all kinds of stuff they incidentally ingest, so that's sort of one of the cool parts of doing this work, is actually getting to stick your arm into the stomach of these fish to see what they've been eating. And one really impressive thing about the spawning aggregations and the research that we're doing and tagging them, we put external tags in them in addition to acoustic tags, and we're able to track these fish over multiple years. Some of the acoustic tags have shown that we'll get some of the same years over an entire year period, or multiple returns within a couple of years. We've caught a lot of fish that we've tagged externally in multiple years, and that tells us a lot about their age and growth patterns and other things throughout over time, which is really cool and not often done. And then probably lastly, I want to talk just briefly about sort of the next phase of my research. And so I talked a lot about some of these measurements that I've been doing. And so really what this comes down to is sort of looking at these measurements using different technologies and studying their isotopes and trying to figure out what, how, and where they're getting this mercury from. So sort of the next phases are to look at mercury isotopic fractionation, which is a technique where I look at mercury in its different forms, different isotopes, which gives sort of a signature of each fish and where it came from. And so comparing fish that I catch in different areas and catching them during the spawning sites will give me some sort of a signature of the areas where they're coming from, which I can then look at its relationship with how much mercury they're getting into their system. So that's one of the big next moves, looking at whether or not mercury's in their gonads, so measuring their eggs and their sperm to see if that's one way that they're off-putting or offloading mercury and trying to get it out of their bodies. Blood, I haven't analyzed any of the blood that I've collected yet. I know, blood has a much shorter residence time in the body, so it's more likely to be a signature of what's happening in the actual area that we're catching these fish from. And so that will be very telling and compare, or in addition to the muscle or fillet tissue and livers that I'm collecting and interpreting for mercury concentration. And so those are the major things. And so collecting these things in different areas will be a future direction, and also over the next couple of years to see how things sort of change from year to year, and in hopes of catching some of the same fish as we've done in the past, and seeing if mercury concentrations in the same individuals have changed from year to year would be really beneficial to the study. So I'm really hoping to see some of that. All right, I'm not seeing a lot of new questions coming in, so either I've rambled too much or people have exhausted themselves with questions. I'm really happy with this turnout, and I really, again, want to thank Teens for Oceans and Explore.org for all the work they do. I'm really happy to be a part of it, and I hope we can continue this thing into the future. I wrote a blog for them that I called Part 1, or Phase 1, because I'm hoping that as I continue this research and discover more things, figure out how mercury is impacting this population and their potential recovery, that I'll be able to report on that. And I hope to keep doing that and potentially have more of these live chats to keep you updated on the research that's being done, and which I think is really cool. And I really appreciate those of you that have listened to this and have chimed in with questions. It's been really nice. So yeah, thank you very much. And I guess with that, I will say goodbye and sign out. So thank you.